today we're actually going to be focusing exclusively on electromagnetic transport or wave propagation. And so, within the context of biomedical optics, this is most pervasive in development of microscopy methods. And of course, microscopy has an ancient, origin. It's been done for many, many years, but particularly in the last 20 years or so, the advent of short pulse lasers has enabled us to deliver very high intensities of light at fairly significant depths from a biological perspective within tissues. And this has enabled a broad variety of linear and nonlinear methods to probe optical processes within biological media which are specific to their electronic and vibrational structures. And this is very exciting because now you can imagine looking at structure and function of tissues without the use of exogenous agents, but using the endogenous composition and structure of tissues to reveal themselves by leveraging various linear and nonlinear processes. And so to give you an idea of this kind of exciting area of research and development, I'm happy to introduce Dr. Mihaela Balu, who's been a staff researcher at the Laser Institute for about 7 or 8 years now, right?
8 years, yeah.
She's a native of Romania and did her undergraduate work there in physics, and then came to the University of Central Florida where she got her PhD in optics. And she's been really have a laser focus on translating these technologies into the clinic so that we can really leverage their power to try to address some very relevant clinical problems and uncover some interesting biological processes using these methods. So let's welcome Dr. Balu.
Thank you. Thank you, guys. Thank you and welcome. So I work at the Beckman Laser Institute at the medical clinic where you guys visited yesterday, I guess. I work in the nonlinear optical microscopy lab there, and we'll visit our labs this afternoon, right? So we'll talk today about optical microscopy in turbid tissues. And let's start with our teaching objectives. We'll learn about this technique that's called laser scanning microscopy, and we'll learn that the focal volume is the defining feature of this technique. How many of you are familiar with this technique? I'm curious, so I'll know. Have you worked with? Okay. Okay. Linear or non-linear? Confocal?
Yeah.
Okay. Okay, well, we'll see. So we'll learn about scattering and how it affects resolution, contrast, and penetration depth. We'll learn that the nonlinear optical microscopy is good for tissue imaging, and that signals generated in coherent techniques are complex. Imaging turbid tissues with conventional optical microscopy. I like to make sure that we understand the answer to the question, why do we need to use optical microscopy to image turbid tissues? Why don't we image cells in a dish to learn about tissues? And that helps to a certain extent. But we do want to get information about the tissue at the microscopic level, because we want to learn about the tissue functionality, for example, to learn about cellular metabolism in the tissue environment. We want to diagnose diseases. We want to be able to do live pathology, to image the tissue and get information about it without taking a biopsy. Or we want to be able to monitor different treatments pathways. So there are many reasons we we want to do optical microscopy in turbid tissues. That would really get us to the point where we can image living tissues. Now, tissues are thick and they have very little contrast. The only way we can image it at different depth using conventional optical microscopy is by sectioning the tissue, by mounting these slices on covers on microscope slides, staining them, and analyze them using a microscope. Really analyze them slice by slice. This is how what the pathologist does. So there are two limitations. There are two problems with this approach, and one is that it's time consuming, and there are many steps involved in the process, many things that can go wrong. And second, we need to really take the tissue out of its own environment, and while the information about the morphological changes are preserved, we lose the information about the tissue functionality, about the cellular metabolism in the tissue. So really the question is, how can we image thin slices of tissue by optical means without having to physically section it? And the answer to that is we do that by focusing the light into the tissue. We focus the light to get information about the microscopic scale, to get to the microscopic scale, and then we scan this focal point across the specimen. So that gave rise to a technique that was originally called confocal point scanning, and later on laser scanning microscopy, and we'll see why. So in wide field illumination, in conventional optical microscopy, each plane is evenly illuminated, and the image of the entire field of view is the entire field of view is imaged on a CCD camera at once. While in this technique, we get an image of the focal point, and then to construct of a 2D image, we need a scanning mechanism to scan in the X and the Y direction, to scan this focal point across the specimen, and then we collect the signal with sensitive detectors, usually photomultiplier tubes. Now, the most important feature of this technique is the focal point, and we want to have a tight focal point to get a good spatial resolution. Now, originally, when this technique was pioneered in the 50s, the white light sources, the white illumination, the broad source illumination was proposed, but then it really took off when the lasers were used as light sources, and that's because lasers are coherent sources. They are bright, they have low noise. We can focus them tight in the specimen. So that's the laser scanning microscopy. Let's see what confocal means. Confocal, really, what's important about confocal is really using this pinhole in front of the detector. Let's see why. So we use a laser that's focused on the specimen, and then the signal is generated in the entire focal volume, in a large focal volume. Now, with this focal volume is imaged on the plane of the pinhole that's placed in front of the PMT, and the purpose, the role of this pinhole is to reject the out of focus fluorescence. As we close this pinhole, we really collect the signal only from the vicinity of the focal point, and that really generates the Z resolution. It really creates this optical sectioning that's important in the confocal. Now this technique can work in the reflectance mode when the laser the wavelength of the excitation is the same as the wavelength of the emission. And in this, so we have a beam splitter here that directs a portion of the laser light into the sample, and then it collects the signal from the sample. I mean, it transmits the signal from the sample to the detector through the pinhole. The signal that it's detected in this method, it's the scattered signal, the reflected signal from the sample that carries the information about changes in refractive indices among different components of the specimen. So this technique is largely used in skin imaging. These are examples of optical sections of human skin obtained with this technique. These are actually images acquired live on the skin of a human subject. It shows optical sections of skin at different depth. We are looking at the epidermal layers this is cellular structure in the skin. These bright cells over here are pigmented cells in the skin, and then deeper here we have the dermis, the fibrillar structures. We don't see it very well in this image. So it can work in the reflectance mode or in the fluorescence mode when where we replace the beam splitter with a dichroic mirror that separates the excitation from the emission. So the fluorescence is generated in a large focal volume, but only the one in the close vicinity of the focal point is transmitted through is collected through the pinhole. And I chose a couple of images of glioblastoma spheroids to show the performance of confocal images compared to wide field images. These glioblastoma are brain cancer cells, and these spheroids are 3D structures. So we really see the benefit of the confocal when we image through these 3D structures. So it's in this kind of images where we see the beauty of the technique, the way it's able to provide this higher resolution images as we section through the tissue. Is it? Yes.
So I have a question a little bit about not the fluorescence microscopy, but just the simple reflectance.
The reflectance. Okay.
So I'm just wondering about the efficiency of throughput in this system, right? Because since your reflected light is at the same wavelength as your excitation, so I would assume if it's a standard beam splitter, you lose 50%.
Yeah, that's true.
Going down, and you lose another 50% of the reflected light going back. Is that true?
Yes, that's true, but that's not the main problem, because to be honest, I'm familiar with this particular application. I don't know exactly where else this. So for skin, you don't want to use too high of a power. You need low power. So the fact that there is some significant loss by the beam splitter here is not that critical. For other applications, it might be, but usually it's not. It's still quite a superficial technique, right? It images shallow. I mean, the fluorophores help. You can get deeper with the fluorescence, but with the reflectance, you can go that deep, especially in this scattering tissues like skin.
So you're not source power limited in any way?
No. So yeah, we should be fine. Yeah, there is another. Yeah.
Can you explain again how you select the depth with this technique?
With this pinhole over here.
Yeah. How exactly does that work?
So this one, the important part is that this is in the conjugated plane with a focal point, right? So when you close this pinhole, you select the amount of signal that you collect. It's really if you follow I try to show the intensity beam distribution. So by closing the pinhole, you really collect the light, the signal close to the focal point. When you open it, you pretty much collect what was generated in the entire focal volume. So yeah, yes.
So does that mean that the lens that you use to focus
Yes.
the incident light into the sample is identical, perhaps, to the lens that focuses the light into the hole?
You really.
That distance is kind of conserved. So to look at deeper depths, do you translate the tissue up? Or do you move that lens
Well, I
down?
think it matters the fact that this is on a collimated beam. You keep the imaging condition as you translate the lens. But yeah, if it's easier to understand, you can think about it as moving the specimen. But again, this is currently applying skin imaging. We don't want to move the patient up and down. We move the lens. Other questions? Okay, we'll move on. So I was mentioning the improved spatial resolution with this technique, and in order to understand about resolution and other aspects of these technologies, we really need to understand the image formation, how the image is formed in a microscope, and really to understand the image of how the image is formed by a microscope, we need to understand how the microscope treats a point source. Why is that? It is because of the Huygens principle. Huygens taught us that all points on a wavefront of light act as point sources for secondary wavelets, and then at a later time, this one stopped working. At a later time, the composite wavefront is the surface which is tangential to all of these wavelengths. So this is how the light propagates. And the important point is that the specimen we are interested in can be represented. The specimen that we are interested in and it's illuminated can be represented as the sum of an infinite set of point sources. Yes, I was about to try that. Okay, thank you. So we can represent any object as the sum of infinite set of point sources. So if we understand how the image of a point source, we understand the image of our specimen of interest. So what's the image of a point source? One might think that a point source is imaged into a point, but I'm sure all of you know that's not the case. In fact, point source is imaged into the image of a point source, it's a blurred point. That's called the point spread function. So why is it a blurred point? That's because the image is the result of the interference, of the light interference of the light waves interference. Light is a wave. Of the light interference in the focal point. So most of the light will goes to the central point of the image, where the waves interfere constructively to form a bright spot, but they can go to a different location where they can shift in phase and interfere destructively to generate a dark point. So the result of this interference is a pattern that's called the Airy disk pattern. It's called the diffraction pattern because the light is diffracted at the objective aperture before it interferes, and it represents the distribution of the point spread function. So it's really this pattern is formed by this bright spot that's surrounded by concentric bright and dark circles, and so this would be the intensity distribution of these patterns. So knowing this intensity profiles of the Airy disks, really it allows us to define and talk about the resolution, which is really so the resolution is defined as the smallest distance between two point-like objects for which their images can still be distinguished. So it's really about the ability to distinguish between two Airy disks. And Rayleigh came up with a criterion for that. He stated that two point sources are resolved when the first minimum of one Airy disk overlaps with the central maximum of a second Airy disk. So if the distance defined in this way, if it's larger, if the Airy disks are farther apart, then the point-like sources are distinguished. Their images are distinguished. If they are closer, these patterns overlapped, and we can't resolve these 2 points. So. Is this clear? Yes.
You use laser from this application. So what wavelength for that?
Well, this is really. It's not particularly for a laser. You can. That's the way we generally define the resolution. So you can see it as the interference pattern of a broader source. So, as we'll see, the resolution does depend on the wavelength. So when we use monochromatic sources, then well, it can apply for any wavelength. We'll see the dependence with a wavelength.
So it's depend on the.
Yes, let me show you. This is where I was going. This is where I was going next. So I just wanted to say that this definition here of the resolution criteria, this is really closely approximated by the point spread function width. So when we measure the resolution of our microscopes, we don't hunt for the central maximum and the first minima. We usually image very tiny objects like small beads, under-resolved beads like some nanometer size, and we image the full width half maximum of the intensity distribution formed by those small objects, and that really provides us with the information about the resolution, and which is the size of the the diameter of these Airy disks, the size of the point spread function. So now that answers your question. This this resolution is really proportional with this factor here. It's directly proportional with the wavelength, and inverse proportional to the numerical aperture. So that shows us that you get better resolution with shorter wavelength. Now I didn't introduce the numerical aperture, but that's defined by the angle of the illumination, or in this case for the white illumination, the angle of the collection. So the larger this angle, the larger the numerical aperture, so the better resolution we get. So that's the relationship between the resolution and the wavelength. Does that answer your question?
When you try to see the tissue under the skin.
Yes.
So what wavelength do you use to see under skin? Because the skin have a absorption.
Yeah.
Scattering.
Yeah.
So,
Well.
different wavelength, you have a different deep.
Yes. Yeah, and we'll get to that. We usually use near infrared wavelength too. Yeah. Yeah, I don't want to anticipate, but we'll talk about that.
Could you just go back one slide? I wanted to set it there.
Okay. Yeah, these are very nice cartoons that I get from this website. These websites of Olympus and Nikon. They have very nice self-explanatory images about the way the image is formed in the microscope.
So the interference really arises from the lens, the presence of the lens, going through traveling different pathways.
Yeah, and the diffraction of on the lens aperture.
Right.
Yeah, that's yeah. Okay, so another thing I wanted to mention. So we have different. Different factors here. For the wide field, for the confocal, the resolution was measured to be lower. No matter what, this dependence on the wavelength and numerical aperture is the same. So that's something you would need to keep in mind for microscopy. So with the confocal, we get lower resolution in the XY direction, so lateral resolution, but also axial. When I was mentioning about the way we measure the resolution, that we image these under-resolved beads. We image them in one plane, in a horizontal plane, but we also scan through them in the z direction to measure the axial resolution. And if we look at the difference between the axial resolution between the wide field and the confocal, we notice the dramatic reduction in the intensity of the wings for this confocal. Now the improvement in the resolution for the confocal is not too it's it's not an extraordinary improvement in the resolution lateral and axial, but what really matters for the confocal is this, it's really the capability of the technique to do this optical sectioning, and that really improves the effective Z resolution. And that's again because of it's the result of this, the integration of the intensity point spread function. It has a maximum when we evaluate it with depth for the in the confocal case. So we get the result of this integral is non-zero on a small depth of about 1 micron, while for the wide field microscopy, you really get no depth dependence of the total intensity, and then the signal from each plane contributes equally to the image, and we cannot ignore the out of focus fluorescence. Now that's an important part. I want to talk about the relationship between interference, coherence, and scattering. We mentioned how important this focal point is when we focus the light into the specimen. Well, it's important to focus the light tight, but it's also important to maintain this tight focusing during the light propagation in the specimen. And I like us to go over the factors that affect this size of the focal point as the light propagates in the specimen. So let's go back to look more carefully at the interference. We said that the image is the result of the interference of the light waves. This is the result of the interference of two plane monochromatic waves. This is called the interference factor, and this is the phase, and it's related to the wave numbers of the two individual waves. That's related to the wavelength. Then the frequency of the two waves, and the phase shift between the two waves that interfere. Now these waves are coherent if they have a constant phase difference over time. So this phase needs to be coherent over time in this way to call the waves coherent. If this phase varies in a random fashion over time, like here, the waves are incoherent. Now these are two extreme cases. In real life, we deal with something in between, with a partial coherence, even for lasers. Now another thing I wanted to mention, this term over here is related to the spatial coherence. That tells us how collimated the beam is, how non-divergent the beam is. This is related to what's called temporal coherence, and it tells us how monochromatic the light source is. Now, an important thing to know is that the phase of the light in the tightly focused beam is distorted by scattering, and the amplitude as well. But I wanted to focus on the phase now. So the scattering distorts the phase coherence of light, and that really means that the waves lose their ability to interfere, and then the focus gets aberrated, gets enlarged. We cannot keep the tight focus if the waves won't be able to interfere. So the coherence really measures the ability of light to produce interference, and so it's very important to know the relationship between them. Go ahead.
Just a point of question / clarification. When you say the scattering is affecting the phase, you mean because the path length of the photons is different depending on the number of scattering events there are? Because, and the reason I ask is because for something like laser speckle imaging, we count on, that there being coherent light that makes it back to the detector, right? That even though they go through multiple scattering interactions, it still retains its phase. But in this case, it's probably because you have different numbers of scattering events, right? Causing each wave that comes back to have a little slightly different phase.
Yeah. You know what I learned? I'd like to learn more about that too. So I think I'll listen to these coming lectures. I like to understand better the relationship between the scattering and the way it distorts the phase.
I don't know, Vasu, do you have any input on that? Because we kind of touched on that yesterday a little bit when we started talking about scattering and.
Let's hold that for now.
Yeah. When we get to diffuse optics, it'll come up probably some more.
The next lecture will also speak to
the issue. So that's really, I wanted to make it as an introduction to the next one. So let's answer these questions later. So we are ready to move to contrast mechanisms that we use in laser scanning microscopy. So if you have other questions for now, are we okay? Okay. So we talked about the contrast mechanism in the reflectance confocal microscopy. So the contrast mechanism in this technique is based on the variation in the refractive indices among components in the tissue. We mentioned the fluorescence confocal microscopy, and the contrast mechanism in this technique is based on one photon excitation fluorescence, and we'll talk about that. We didn't talk at all about nonlinear optical microscopy, but we will. It's based on a bunch of nonlinear optical signals, and we'll talk more about three of them: the two-photon excited fluorescence, second harmonic generation, and coherent anti-Stokes Raman scattering. Let's start with the fluorescence, which is the property of some atoms and molecules to absorb light at a particular wavelength and emit light of longer wavelength after a brief interval. So in this technique, this is it's called the Jablonski energy diagram to show exactly how this process occurs by using the energy state. These are electronic energy states, and the gray lights are our vibrational excited states. So in this process, an incident photon is absorbed, and it excites the molecules on a higher energy state. Then some of the energy is lost through a non-radiative vibrational relaxation, and then a fluorescent photon of lower energy is emitted. So it's important to keep in mind that the fluorescent photon has lower energy. Lower energy means a longer wavelength. So if you have if you excite the molecules, let's see in with a 400 nanometer, we expect the fluorescence spectrum to correspond to longer wavelength to the right of the spectrum. The fluorescence spectrum is you expect a fluorescence spectrum to be broad because of the multitude of the vibrational states involved in the process. So it's a linear excitation, which means that each absorbed excitation photon produces a single emitted photon. And these are some origins of fluorescence contrast. We use labels, which are free dyes or nanoparticles that really they attach to different components in the tissue. You can tag, you can label the nucleus or the cytoplasm or some specific. Oh, go ahead. Thank you.
Can you go back to the slide before?
Okay.
Hopefully I'm not asking too much.
Sure.
Talk a little bit about the phosphorescence case.
Yeah, that's a different pathway that it can take. It's also a radiative emission. It happens at a different lifetime. The lifetimes of the emission is different. So I'm sorry, it was on the diagram here. I just didn't touch on it because we are not going to talk about phosphorescence, but it's just a different pathway in the emission. Yeah, so we have labels that are free dyes which partition to a specific region of the cell. We can have indicator dyes that are probes that bind an ion and then they change their fluorescence emission or excitation spectrum. We have fluorescent proteins such as green fluorescence protein that are produced by the organism after the DNA for GFP is introduced into the cell. So very importantly, these are exogenous dyes. These are external agents that you add to the tissue to be able to light up different components in the tissue, like nuclei or cytoplasm or some fibrillar structures, really what you're interested in imaging. We also have intrinsic fluorophores that are naturally occurring fluorescent molecules and proteins in the system. And examples of that are NADH, FAD+. These are two coenzymes in the mitochondria of the cells with a very important role in the cellular metabolism. And also we have naturally occurring fluorescence from collagen and elastin, and these are proteins in our in the connective tissues. And this is an example of how these fluorophores work, these exogenous fluorophores. And again, I chose images that would show the difference. Now, can we have the lights off? Yeah, I think it would help. Oh, that's good. That's good enough. Thank you. So these images really show the difference in the high resolution that we get when we do this optical sectioning, when we look deeper in the tissue. These are all thick sections. This is brain, this is muscle, this is a sunflower pollen grain. So these structures are tagged with different fluorescent dyes. And so we can visualize them individually. And when you are interested to image multiple fluorophores, you can do that by splitting the excitation. By splitting the emission with using different dichroic mirrors. So you add one or more dichroic mirror depending on how many fluorophores you want to detect, and then you use detectors and really detect the signal in different spectral windows, and then you overlap the detection channels to get this kind of images. And yes. Go back? Yeah.
So if you have more than two fluorophores that you want to
Yes.
So how would you, I guess, how would the technical, so you would use, I guess, let's say you want to capture 4, so you use 2 dichroic to do that? Or how, I guess, how would
Yeah, it depends on the number of the fluorophores. If you have 2 fluorophores, you use 1 dichroic mirror. If you have 3 fluorophores, you use 1 more dichroic mirror. So you split, let's say the shorter wavelengths, you leave the longer wavelengths pass, and then you add another dichroic and split farther, and split the emission signal farther away.
So depending on the number of fluorophores, the way you arrange the dichroic mirrors and the ones you actually use will change.
Exactly. Yeah. So it's a lot of it's really important to know the information about your fluorophores to be able to choose the right optics, the right dichroic mirrors. It's not only dichroic mirrors; these are usually not enough to for the efficiency of the signal collection. You also use filters in front of the PMTs. I didn't add them, so I wouldn't complicate the drawing. But yeah, if you understand your fluorophores and what you are expecting to image, you'll be able to choose the right optics to detect the right signals. Okay. So need to move ahead and introduce the nonlinear optical signals, and to do that, we need to summarize the limitations of the linear fluorescence and of the confocal laser scanning microscopy. So first of them, I mentioned before, the molecular contrast is obtained only through labeling fluorescence for the linear fluorescence, and the reason for that it has to do with I don't know if I want to anticipate. I can explain that later. It has to do with the photo damaging. You use and the fact that the fluorophores, the exogenous dyes that we add to the system that needs to be imaged, they can be excited more efficiently than the endogenous dyes. So for the endogenous dyes, for these fluorophores that we have in the system and they are intrinsically fluorescent, you need to pump them harder to be able to get fluorescence, and this is when you get photo damage. If you use longer the wavelength for that, the likelihood is less, and we'll see that. So also this technique it uses short excitation wavelength, and that provides limited penetration depth. Short excitation wavelength means they are scattered more. So this anticipates the fact that the nonlinear optical microscopy uses long excitation wavelength, so it offers a increased penetration depth. It uses a small aperture detection, which means that the technique requires high excitation power to detect enough photons. And when there is photo damaging, that happens through the entire illumination cone. And as I anticipated, a nonlinear optical microscopy addresses these limitations, and it does that through the confinement. It confines the generated signal, let's say the generated fluorescence, to the focal point. So it doesn't generate the signal in the entire volume as the linear fluorescent does, but the signal is really confined at the focal point. And why is that? It's because the nonlinear optical signal depend nonlinearly on the excitation intensity. And that really means if you think about it, the intensity is the power per unit area. So you can think of it when you focus the beam in the vicinity of the focal point, you get a larger area that in the focus. So that means lower intensity, and then the probability of generating this process decreases abruptly as you depart the focal position because of this nonlinear dependence. And there are many forms of nonlinear signals, and we'll talk about that. That's another way to show how the confinement of the signal at the focal point for the nonlinear microscopy compared to the linear microscopy. This is a classic example. You'll see it often in textbooks and presentations if you get to do microscopy. It shows how the fluorescent is excited in a large cone of light. When you excite the fluorophore, this is a dye in a cuvette. When you excite the fluorophore with the short wavelength, and then how the fluorescence is confined, I'm going to take this off so you can see the dot, the point where the arrow shows. So this is how the excitation occurs when you excite with a long excitation wavelength, and you get this nonlinear fluorescence. Is that clear? No?
I'm not sure how the dot ended up there.
How the dot ended up there?
Yeah.
Did you want to say? Yes.
So Mihaela, will you be talking about the generation of multi-photon fluorescence?
Yes.
In the Jablonski diagram?
Yes, I just wanted to, yeah, do you want me to skip to that?
Well, I think it may explain why you see that.
Yeah, I just wanted to, yeah, absolutely. I can go to that now. I just wanted to really make sure that we understand the benefits of the nonlinear to the when compared with linear.
Well, I don't mean to disrupt the talk. I mean, if you're going to get to that, that's fine.
I will.
So you're just, right now, just to clarify, you're just showing that if you have a nonlinear signal, you have the opportunity to stimulate a nonlinear signal.
At the focal point, yeah.
To only generate light at the focal volume without generating light outside the focal volume.
Yes.
Okay, so that's the point she's trying to make now, and I think she's going to discuss
And
how this comes about in just a second.
And that's because, this is what I was trying to explain, that's because of the nonlinear dependence with the intensity. So the fluorescence that's generated over here, that depends linearly on the intensity. So even if the intensity drops so this is how the excitation looks like in the specimen when you focus it. So even if the intensity drops as you depart the focus here, you still generate some fluorescence because it's a linear dependence, but of the fluorescence with the intensity. But for the nonlinear, when you depart the focus, when you get far from the focus, the total the probability to generate a nonlinear signal is way much lower because it depends let's say it depends with the I squared, with the squared of the intensity. Really, the probability to generate a nonlinear signal depends, is proportional to let's say to the I square. When you get far from the focal point, that probability gets much lower. So you just don't generate the signal farther away from the focus. Go ahead. Yes.
I was asking why the two figures are similar, because because of the nonlinear characteristic of the
Why the two figures are similar?
figure should be, the beam waist should be thinner, isn't it?
Well, yeah, I'm sorry, that's a detail. We should, you are able to focus the short wavelength tighter than the long wavelength. This is what you mean, that I should have adjusted the beam waist?
The size of the laser beam
Yes,
on the focal plane,
yeah.
the nonlinear microscopy should be thinner.
You mean the size of the excitation or the size of the emission?
The beam waist at the focal plane thinner because you have too much power. Isn't it?
No, the excitation they have. I thought you meant the resolution. You can focus the shorter wavelength. You can get better resolution. Remember when we talked about that. So yeah, if you use the same optical arrangement, the same lens, you can focus the shorter wavelength tighter, so it can have a smaller beam waist compared to the. It doesn't have to do with the power.
With the same wavelength. With the same wavelength for both linear and nonlinear microscopy.
You cannot do them with the same wavelength.
I think this will be clarified once you use the Jablonski diagram.
Okay. Yes. Yeah, I should. Next time I'll organize them. Differently. Another thing I wanted to talk about was how this what's the scattering effect on on linear fluorescence as opposed to nonlinear processes. And for that, we need to be clear how what's the effect of scattering in general. So it distorts and it attenuates the excitation light, which means that the focal point will become aberrated, will become enlarged, and then the signal is generated in this enlarged focal point, and then the scattering can propagate the signal out of the tissue. So it affects the excitation, and through the excitation it affects the generated light, and it also affects the emission. Let's see how it affects the confocal, the signal generated in the linear fluorescence in confocal microscopy. We have a high distortion and attenuation of excitation light because of the short wavelength excitation. So I hope that's clear. The shorter wavelength are scattered more than the longer wavelength, right? That's why the usual explanation why the sky is blue. It's the focal volume is enlarged. We talked about that when we explained the relationship between interference, coherence, and the scattering. How the coherence phase is affected, and then the waves lose the ability to interfere, and then the confocal detection sees only the ballistic photons because of the confocal arrangement. So it loses some the photons that are scattered away and are not detected. So it's not such an efficient way of detection. In nonlinear microscopy, we have a lower distortion and attenuation of the excitation light because of the long wavelength excitation. the focal volume gets eventually it gets enlarged, although the distortion is lower as the light propagates into the tissue. The focal volume can get enlarged, and emission is generated in this larger focal volume. But now we have an open aperture detection. We don't need to use this confocal pinhole, and then that sees the ballistic and non-ballistic photons from the entire excitation volume. So again, that means the scattered and the non-scattered photons. And now one more thing before we go to the Jablonski diagram for the 2 photon. I just wanted to summarize again the differences between these two, the nonlinear optical microscopy and linear microscopy. With nonlinear optical microscopy, we can use the molecular contrast of endogenous molecules, and that this really opens the way of to image living tissues, as opposed to linear microscopy when the contrast is obtained only through labeling. We talk about increased penetration depth due to long wavelength excitation, intrinsic 3D resolution because of the nonlinear dependence of the intensity. We have wide aperture detection, so we collect many photons. They are more photons than in the linear microscopy, so it's really a more efficient way of detecting. And then we get photo damaging only in focus. Let me get to this table later. I want to explain later why is this more expensive than the linear is because of the lasers that we use. So let's get to these processes and explain the nonlinear. What exactly does it mean? So we have many nonlinear optical processes that we use. We use them to light up different components in the tissue. We use coherent anti-Stokes Raman scattering to visualize lipids and proteins, second harmonic generation to visualize collagen fibers, fluorescence to visualize other components. I'm going to talk about three of them. Hopefully we'll have time. But before that, so let's try to understand what nonlinear means. Light interaction with matter. Light is an electromagnetic wave. It means it has an electric and a magnetic component. When the electric component of the field of the light, when it interacts with the charges, the charges in the molecules of the material that the light is incident on, then it polarizes them, it displaces these charges from their equilibrium position, and it induces a dipole moment. The dipole moment per unit volume is called the polarization, and in the linear regime, this polarization is proportional to the electric field. Also, we have a restoring force that's proportional to the displacement. It's called the Hooke's law, and the potential energy that's associated with this restoring force. It's proportional to this force and the displacement, so it has a quadratic dependence. I can. So this one summarizes the linear regime, what I just said. So in this linear regime, the polarization has the same, it oscillates at the same frequency as the driving field. When we use a higher power light, we have a nonlinear interaction in the sense that this restoring force it's not it's no longer it no longer has a linear dependence. It has a nonlinear dependence with this displacement, and consequently the potential energy will won't have a quadratic dependence, but a higher nonlinear order. Then the polarization is no longer linear, and then the resulting polarization will oscillate at a different frequency than the driving field, and that shows that there are other frequency components in this polarization, such as I'm sorry, that gets me first to the second harmonic generation, and then I'll go to the two-photon fluorescence and the Jablonski diagram. So in this process, the resulting polarization, we can decompose it in one component that's the same as it has the same frequency as the driving field, and then another component that has a frequency that's twice the frequency of the driving field. Now, when we have a higher order nonlinearity, that these components keep adding up, and then we'll have a more complex form of the polarization. It's the polarization for this process for the second harmonic generation is non-zero only for certain kind of materials. They are called non-centrosymmetric, and I'll give some examples of what these materials mean. It's really a process where two photons interact through a virtual state. Please keep in mind that this is not an absorption process. There's no absorption involved, so it is just the interaction of these two photons, and then one photon at twice the frequency is generated. So twice the frequency means half the wavelength. If we use an incident light, an excitation light of 800 nanometer, we expect the second harmonic generation light at 400 nanometer. And let me show you some examples of biomaterials that are non-centrosymmetric. One of them is collagen type 1. That's an important protein that we image in the connective tissues. We have the microtubules that are components of the cellular cytoplasm, and we have myosin in muscles that we can image by using second harmonic generation. And that gets me to the Jablonski diagram of the two-photon fluorescence, where we have so in this process we have a simultaneous absorption of two photons. The molecules gets excited on a higher energy state. Again, there is some loss in energy through non-radiative vibrational relaxation, and then a photon is emitted that has an energy that is less than twice the energy of each of these incident photons. So that really means that the wavelength of the emitted photon is shorter than the wavelength of the excitation. If we compare with the linear process, again, if you excite the same molecules with 400 nanometer, you expect the emission spectrum to be on the right of the spectrum in the linear case. If you excite them with 800 nanometers, so these are so imagine you have molecules that, and these are the energy states that correspond to that molecule. You can excite them with one photon, so you would have one photon that gets the molecule excited from this level to this level, and that corresponds to this excitation. And then the fluorescence would have less energy, so a longer wavelength. And now you, instead of using one photon, you use two simultaneous photons to get the molecule on the same state. And then the fluorescence is less than twice the energy of this. And then so the wavelength, the corresponding wavelength, would be shorter than the wavelength of the excitation. So in this case, the two photon would produce a single emitted photon, and that's. Do you have a question, Basan? Oh, okay.
So two questions. So when you're exciting twice for two photons, do they always have to be the same wavelength? Or can you excite with two different wavelengths?
Actually, you can excite with two different wavelengths. That's called non-degenerate fluorescence. You can do that. But usually, when I was talking about two photon excited fluorescence, that implies that it's a degenerate process. It implies that the wavelength of the two photons are the same. We usually, with that, we use one beam. In your case, you need to use two beams, and really the important part is to overlap them both spatially and temporally at the sample location to get the process, because it needs to take one photon, each photon from each beam. It needs to absorb them in the same time for the process to happen.
How is the timing achieved with the excitation? How do you get the first photon and the second photon? How do you actually get that?
Oh, these are. Please don't get confused with what I said before. For this one, you use a single beam. So the photons are absorbed from the same beam, and this is they are absorbed simultaneously in a very short amount of time. That's on the femtosecond scale. If you think about it, femtosecond means 10 to the minus 15 seconds. That's really simultaneously. Yes.
So let me maybe.
Yeah, you can, if you can
explain. Also, perhaps clarify. So,
Okay.
So traditionally, two-photon fluorescence, is described as a process that is, the excitation is linked to the square of the intensity. And the square of the intensity arises directly from the probability of having two absorption events simultaneously.
Mm-hmm.
In order to have the probability of having two absorption events simultaneously is directly proportional to the intensity square. And so that's why, and that is then linked to the spatial confinement you get in the focal volume.
Yeah. I just hoped I explained that well. If I didn't, I'm willing to talk to you more about it. How the nonlinearity dependence really gets you to the confinement. I think that was the part that was not very clear to you guys. But
do that is if you plotted just in traditional Gaussian
Yeah.
propagation, what is the intensity as a function of z along the beam propagation, and then if you hit that with the square, you'll see it.
Yeah.
Can you tell us a little bit more about what you mean by simultaneous, and what gives you? You said that it has to be within a few femtoseconds. Where did you pull that number femtosecond from? Is that linked to Heisenberg uncertainty principle? Is it?
It's linked to but please don't have me get them into that. I think it's really. I think that would require a few more, some more extended explanation.
Any more questions?
I really wanted to be able to talk about Raman as well, so I'd like to move forward. I have only 10 more minutes left. Yes.
I think it's important, because actually it was unclear to me, the difference between second harmonic generation and two-photon excited fluorescence. So two-photon doesn't require having that non-centrosymmetric material, is it?
It requires non-centrosymmetric material.
The two-photon does?
Oh, no, I'm sorry, the second harmonic. Yeah. No, the two-photon, no, it's an absorption process. It's a completely different process. It has a different,
Which I think, which was unclear to me. It's an important point. It's two completely different set of physics, basically.
Yeah, absolutely.
So does any material that exhibits linear fluorescence, can that have a two-photon signal? Or is there something?
Oh, yeah, especially the fluorophores, the labels that we usually use for to label, different biological components in specimens. Those can be excited with two-photon. Not most of them, I would say. Again, it's another set of another discussion we would have on different selection rules and what makes some molecules being excited by one photon and not by two photon. But most of them that we use, you can excite them with both. That really gets me to the next slide, where I wanted to show you what biomaterials are good targets for two-photon excited fluorescence, and these are more commonly the endogenous fluorophores in the tissue. One of them is the elastin, another protein in the connective tissue. That's an image that shows these elastin fibers in green. We get two-photon excited fluorescence from this NADH component in the mitochondria of the cells. These are images of cells in our skin, and we get two-photon excited fluorescence from melanin. That's the pigment of our skin, and these are again images of pigmented cells in the skin. It's very highly fluorescent, and we can also combine these two detection channels, the two-photon excited fluorescence and SHG. Similarly, as I told you with the confocal, when you split the emission with dichroic mirrors, so imagine you do the excitation with the 800 nanometers, you detect the second harmonic at 400 nanometers, you have a dichroic that separates this wavelength from the rest of the fluorescent wavelength. So that's an image of the skin. We can get it's actually a vertical section through the skin that shows the top layer of the skin, the epidermis, and then this is the dermis. In the epidermis, we get fluorescence from cells through the NADH, through the keratin in the cells that's also fluorescent, and through the melanin. And then we image the collagen with the second harmonic generation. It's blue in this image, and then the fluorescence from the elastin fibers in the dermis. Another thing. Another issue I wanted to point out is the fact how we lose the signal when the light penetrates deeper in this scattering tissue. Go ahead. Did you have a question?
How different from the top?
Yeah, it's this. That's a good question. This is what I wanted to talk about. This is the scale bar here is about 20 microns, so it's about 120, 150 microns. So this is how deep we can image in skin with this techniques. And that's because of the scattering. That's because the scattering really affects the focal point of the excitation. So we start with this tight focal point that gets enlarged as it propagates in the tissue. We can compensate for that by increasing the power to one point, but then the scattering also affects the emission. That's the scattered photons do not go back on the same on this pathway to be collected. They are scattered. So we really lose signal due to the scattering as the light penetrates deeper in such turbid media.
When you say this one with 800 nanometer wavelength.
Yes, you'll get to see. I'll do a live demo of the way we acquire these images when you'll visit our lab this afternoon. So I can answer more questions then about it.
Deeper than 100.
It really depends. It really, that's an approximate number. It really depends on the skin type we have. We are lighter or darker, or we can penetrate deeper in the lighter skin when we don't have the absorption from melanin, from the pigment in our skin. We usually image different lesions. We image skin cancer, so it depends on different lesion and how scattering it is. But that's pretty much a typical depth that we can penetrate in the skin. Go ahead.
So Mihaela, if I look at this image,
Yes.
and I'm making interpretations in my mind which I want to know whether are correct. I mean, if I look closely, the bottom 20 microns, you start to see a blurring or a loss of resolution. I mean, you see a dimming, but you also see at least these fine fibrillar features are
Yes.
not only dim, but they're blurred. At
Yes.
least in my mind, they are.
Yeah.
And so that seems perhaps to be due to the fact that you are losing the spatial confinement of your being.
Yeah.
Perhaps. The other thing that I'm seeing is that if I look at this structure, say, relative to this other structure on the left, here it looks like that the resolution is degraded more so here than here. And that could be because something is happening up here that's different than's happening up here, meaning that there may be locally some more scattering up here, which is preventing the confinement a little worse in this portion of the sample than this. So it's not only it's not like the resolution cuts off uniformly across the whole sample, but it's actually spatially
That's
varying.
very true, yeah.
Because it's accumulation of scattering along the whole axis of the beam propagation.
Yeah, especially for this application of skin imaging, where the skin is very non-uniformly and very undulated, especially at the junction, and then on the top we have these lines, not necessarily wrinkles, but really lines that different refractive indices. It acts differently on the beam in different locations. Yes.
And in the right side, you've lost signal completely.
Yeah,
right?
yeah,
And that's
yeah.
not due to an optical design. That's because the tissue has
Yes.
extinguished the light
Yes.
above it completely. So.
Yeah. That's very true. Yes, we have one more question.
I was wondering if you knew if anybody has been using like combination of like nonlinear with like 4 pi. I don't know if you've heard of like 4 pi.
I don't know exactly. I
like
don't.
Microscopes.
Yeah.
To gather the signal.
Do you know about that, Basan? I'm not sure about this
I
combination.
don't know about 4 pi per se, but I do know that in the laboratory for fluorescence dynamics, which is actually on this floor in this building, it's the lab headed by Enrique Corton. They do a method of deep tissue imaging where typically, and correct me if I'm wrong, Mihaela, this system, the objective that focuses the beam is also the objective that collects the photons.
Yes.
What Professor Gratonen, who's
group are doing is that they actually detect the fluorescence in transmission, but they do it by having a huge detector.
Exactly, yeah.
that scans the
So
entire
imagine that
diagonal.
over here, down here, you have a large detector that's really able to capture all the scatter that we miss when we collect with the same
Basically
objective.
collects every single fluorescent photon that is transmitted through the tissue. And so you are able to, your signal generation is higher, and actually you can actually collect generate images very deep.
Is that with nonlinear or no?
Yeah.
Yeah, with nonlinear. The only drawback to that is that it's quite limited for the in vivo imaging, right? You can't quite image on patients with that. But they've been working, as far as I know, on mice models where they pull the skin or whatever they are imaging, an internal organ, and really, squeeze this specimen in between the detection and the delivery light optics. Yeah, Tom?
You mentioned earlier that collagen autofluorescence. So can you get both a second harmonic generation and a two-photon signal from collagen?
Yeah, for particular wavelengths. Somehow you get more fluorescence as you tune the wavelength lower than 800 nanometer. Above 800 nanometer, you don't get much fluorescence from collagen, but you get for wavelengths such 760, 780. We have a nice publication on that. Not we, I mean people in our lab before me, of how the fluorescence signal from collagen depends on the excitation wavelength. So, yes.
I guess with it being in either reflections or transmission mode, so I understand that when fluorescence happens, the photon gets absorbed and the emitting photon can go in any direction. But for second harmonic generation, is that the case, or is it
No, that's a coherent. I'm sorry, I just didn't have time to touch on all the aspects of this emitted signal. That's a coherent signal. The phase is really dictated by the fundamental, by the incident, the phase of the incident light. So that's it has a well-defined direction of propagation. It propagates in the forward direction, but then what we collect in the backward direction, we usually collect the photons that are scattered back. So you can collect both in the forward and the epi direction. I'm sorry, I won't have time to go over the Raman, but at one point I had you see, I had this is a typical layout of a microscopy that's for the Raman, for the coherent anti-Stokes Raman spectroscopy. But this is how we use the detection system in our microscope. So we have these epi detectors. This is how we call it when we collect the photon scattered back. So imagine this. Here is the objective that focuses the light on the specimen that sits on the stage here, and we use detectors to collect in the photon scattered in the back direction, and we also have detectors that collect the signal in the forward direction for signals such second harmonic generation and cars that are coherent signals, and they are propagating in the forward direction. But now again we use the forward detection when we image thin sections. If we image if we do an in vivo animal imaging, or so when the forward light is really blocked, we need to use the photons that are scattered back. Is that okay? Yeah, I'm sorry. I think I'll go to the summary. Well, we'll talk more about the other. I'll try to explain the third nonlinear signal that I wanted to go through. I'll do it when you'll visit our lab, because we have the cars microscopy on the table in the lab. So I'll be able to explain more over there. Yeah, these are another images that I had as applications. I wanted to really to end up by mentioning again that the scattering attenuates and distorts the excitation volume, and is the main limiting factor for penetration depth in microscopy. And we would like to know if this can be better understand, if the scattering effects can be reduced in any way, and if the penetration depth and the image quality can be improved. And I hope you'll get some knowledge about that from the next lectures. And then I'll finish by summarizing again our teaching objectives. We learned about the focal volume, that it's the defining feature in the laser scanning microscopy. We learned how the scattering affects the resolution, the contrast, and the penetration depth. We learned, hopefully, that the nonlinear optical microscopy is good for tissue imaging. And I'm sorry I didn't get to show you the most complex I was planning to show in terms of nonlinear optical signal, which is the Raman signal. But I'll tell you about it as you visit our lab. And thank you very much.
Any further questions for Michaela?
Could you maybe quickly comment on the size and expense of the laser that's required to do these techniques?
Oh yeah, I wanted to mention that. So we do the nonlinear optical microscopy. We do that with mode lock laser with short pulses, and we do that because we want to have we need a large flux of photons at the focal point, and for that we use high peak power. High peak power means, so power is energy per time. You can get high peak power by increasing the energy or by shortening the time. With the short pulses, we do that by by delivering light through short bursts of energy. We don't want to work with high energy because of photo damaging. So it requires short pulses. We use femtosecond and picosecond pulses, and these lasers are expensive, more expensive than the CW lasers that people use for the reflectance or fluorescence confocal microscopy. So yeah, it is a complex and an expensive technique to use, but it's worth it, I would say, because I work with it. Any other questions? Okay.
Okay.
I think
we'll have a break till 10:30.
Oh, I took time from the break. I thought I took time from the next lecture. Okay, even worse. Thank you.